At Cary Academy, in our Advanced Biotechnology class, we've been working on a human disease pre-diagnostic experiment with sickle cell anemia in finding primers to amplify the genes. So far, we have researched and seen that the single gene mutation occurs on the short arm of chromosome 11, p15.4. The change that actually happens in the DNA sequence is a substitution of a T in place of an A. The HBB gene, which is where the mutation occurs, is responsible for the production of hemoglobin, which gives red blood cells their shape. Its primary function in the blood is to carry oxygen. It has 4 subunits, 2 beta and 2 alpha, as seen here in the image. The mutation is located on the beta subunit, and the mutation replaces beta globin, which is the type of hemoglobin that is normally produced for red blood cells. Mutated hemoglobin includes hemoglobin S, C, and E. This one mutation causes the hemoglobin in red blood cells to form abnormally, resulting in the sickle-shaped cell. These cells are generally less flexible and can cause blockage in the bloodstream. The symptoms that are presented in a patient before diagnosis includes pain, fatigue, swelling in the extremities, and loss of vision. A second factor in sickle cell anemia is the presence of thalassemia. This results in a disease called B thalassemia, which reduces the amount of hemoglobin. If one parent has the sickle cell trait, then the offspring, with B thalassemia, could have a condition called sickle B thalassemia, which has the potential to be just as bad as sickle cell anemia. The mutation is inherited from the parents, and there must be 2 copies of the gene in order for a person to have actual sickle cell disease, making the mutation recessive. Only 1 copy, or being heterozygous, results in a sickle cell trait. By itself, having the trait won't affect the patient, but if paired with thalassemia, the patient could experience symptoms similar to sickle cell disease. Despite its negative effects, sickle cell has provided those affected by an immunity to malaria. Malaria is a disease that affects the blood cells and can cause blockage in the capillaries to the brain. Having the sickle cell trait is the best case scenario in terms of prevention against malaria. In this case, a person wouldn't have sickle cell symptoms but they would also have a 60% protection against overall mortality relating to malaria. If a patient were homozygous for sickle cell anemia, they are more likely to suffer from the deadly effects of malaria. There are some forms of genetic testing for sickle cell to detect the mutated hemoglobin S.
Research was done on the DNA sequence and the mRNA sequence so that primers could be made. The online Mendelian Inheritance in Man entry for hemoglobin beta subunit page was used to get the information that was needed. Ensemble was then used to obtain the cDNA sequence, and the NCBI reference site provided the same information. The DNA accession number was inputted into a nucleotide blast search, and then the NCBI reference sequence was found. That was searched until the full DNA sequence was obtained, along with some of the adjacent genomic DNA. Only the HBB gene is shown in the slide. The protein sequence was also found through Ensemble, along with the protein variants. The final step was to design primers to amplify the gene. The change in the DNA sequence occurs in the first exon of the HBB gene, so primers were designed to contain that area. That area was isolated on the NCBI map viewer feature. After getting the area, the primer blast search tool on NCBI was used and provided 9 primers that would work in that general area. The primers that were selected are shown. The set of primers were selected because they were close to the goal qualifications that were assigned. The length of the primers was within the 18 to 22 nucleotide goal. The TM of the forward and reverse primers were relatively close, and the product of the primers were a good length, which is above 200 and below 1,000 base pairs, so that it could be viewed on an agarose gel. The set of primers isn't perfect, though. Its self-complementary rating is relatively high, and the forward primer's GS percentage isn't close to 50. The primer set was selected was the best that primer map came up with. Primer map was then used to verify that the primers would work and help identify a restriction enzyme that would make a single cut in the middle of the amplified sequence. The primers, according to primer map from bioinformatics.org, would bind at the middle and the end of the desired area. The restriction enzyme that will be used is NCOL. As shown, NCOL should make it cut 425 base pairs into the area target. The next step will be to order the primers that were selected. They were ordered from Thermo Fisher Scientific. The plan is to put genomic DNA with the primers into a PCR machine to amplify that region in the DNA. A restriction enzyme digest will be used with NCOL to see if the cut was in the correct amplicon. This will allow it to be determined if the amplification process worked in the experiment.
To close, we have a couple questions that we were hoping you would be able to answer to help our research progress. Do you have any suggestions in improving our primer locating process? Do you know of any other single gene mutation similar to sickle cell such that it provides immunity for another disease? Do different types of hemoglobin result in different symptoms of sickle cell? How soon will gene therapy be used as a cure for sickle cell? And do you have any knowledge on how qPCR should be used to diagnose PCR?
We look forward to hearing your answers, and thank you for your time.